As you probably know, proteins are the result of a multi-step process. First, the gene coding for the protein is transcribed into a messenger RNA molecule. Second, the messenger RNA molecule is translated into a protein. In many cases, the activity of the newly synthesized protein requires both modifications, called post-translational modifications, and interactions with specific ligands. For example, enzymes depend on the interaction with the substrate. The activity of some enzymes is controlled by allosteric modulators, while transient covalent modifications, such as phosphorylation, can activate or inactivate some enzymes. In this video, you will discover another mechanism of post-translational activation of a group of proteins playing a variety of roles, from digestive enzymes to polypeptide hormones. For example, some enzymes are synthesized as inactive precursors called zymogens or proenzymes. How are zymogens activated? The activation is triggered by an hydrolysis reaction catalyzed by a proteolytic enzyme. The result is usually a change of conformation of a portion of the polypeptide, revealing the active site of the mature, fully processed enzyme. Very often the processing occurs inside the Golgi system, where a specific region of the zymogen is cleaved. The activation can be as simple as the cleavage of a peptide bond in a precursor, or the removal of a short peptide, or even the removal of a larger, independently folded polypeptide exceeding 100 amino acids in length. While these cleaved-off regions inactivate the enzyme, they are in some cases necessary for the stabilization and proper folding of the zymogen. The inactivating peptide cleaved off during the activation of zymogen should not be confused with signal peptides located at the N-terminus of proteins and used to direct proteins to a specific intracellular compartment. As shown on this diagram, newly synthesized proteins have several fates. Some proteins made on free cytoplasmic ribosomes have no signal sequence and will remain cytoplasmic. Other proteins synthesized on free cytoplasmic ribosomes have a specific N-terminal signal sequence that direct these proteins to mitochondria, the nucleus, or other small organelles such as peroxisomes. In some cases, translation is initiated in the cytoplasm, but quickly after the signal sequence has been synthesized, the ribosome attaches to the surface of the endoplasmic reticulum. Translation resumes, and proteins accumulate in the lumen of the endoplasmic reticulum or get embedded in the endoplasmic reticulum membrane. This protein will be transported to the Golgi system and then to secretory vesicles, or to a type of cytoplasmic organelles called lysosome. Proteins contained in secretory vesicles get secreted by cells, while those contained in the membrane of these vesicles will be integrated in the membrane of the cell. The signal peptide is organelle specific, and in most cases cleaved off by protease after delivery. Signaling peptides and inactivating peptides represent a powerful mode of regulation by which a protein can be activated where and when needed. Let's now examine in detail a specific type of zymogen called chymotrypsin. This digestive enzyme is produced and secreted by the pancreas in an inactive form called chymotrypsinogen. In the duodenum, the active chymotrypsin is a proteolytic enzyme that breaks down proteins from ingested food. Chymotrypsin is the processed and active form derived from chymotrypsinogen, an inactive zymogen containing 245 amino acids. The first step of the activation process is the proteolytic cleavage of the peptide bond between amino acids 15 and 16. This reaction is catalyzed by a protease called trypsin. The end product is the active pi chymotrypsin, in which the two cleaved fragments remain covalently attached via a disulfide bond. In the final step of the process, pi chymotrypsin takes over and digests itself. Two dipeptides are cleaved off and then released. The first one contains the residues that occupy position 14 and 15 in pi chymotrypsin, and the second one contains residues that occupy positions 147 and 148. The outcome is the mature form of chymotrypsin, called alpha chymotrypsin, comprised of three polypeptides that remain together because they are linked by disulfide bridges. What does occur at the structural level when chymotrypsinogen is converted into chymotrypsin? The side chain of isoleucine occupying position 16, here shown by a block arrow, points toward the exterior of chymotrypsinogen. As you remember, trypsin cleaves the peptide bond between residues 15 and isoleucine 16. Upon cleavage, isoleucine 16 can pivot and turns inward. Now the free amino group carried by isoleucine 16 can form an ionic bond with aspartate 194. This new electrostatic interaction triggers a series of conformational rearrangements, moving methionine 192 from a deeply buried pocket to the surface of the enzyme. Finally, the movement of this methionine residue leads to a rearrangement of loops in the surrounding area. On this close-up view of the active site of the enzyme, you can observe that the induced conformational changes results in the creation of a hydrophobic pocket at the surface of chymotrypsin. This pocket accommodates the hydrophobic side chains of residues from protein digested by chymotrypsin. Indeed, chymotrypsin cleaves proteins on the C-terminal side of the hydrophobic residues phenylalanine, tryptophan, and tyrosine. As I previously stated, proteolytic activation of proteins is not limited to enzymes. Other proteins, including hormones and neuropeptides, are also activated by proteolysis. Here I'll take the example of insulin. You learn in later videos that insulin is secreted by the pancreas in response to a high blood level of glucose and stimulates glucose uptake by tissues such as liver, adipose tissue, and muscle. We know that insulin is secreted by pancreatic cells, but how is insulin processed? Insulin is a secreted protein, therefore it transits through the endoplasmic reticulum, the Golgi system, and finally secretory vesicles. Insulin is produced as a 110 amino acid precursor called pre-proinsulin. As it is translated at the surface of the endoplasmic reticulum and is pulled through the ER membrane, the N-terminal signal sequence that directs this precursor to the secretion pathway is cleaved off. The result is the accumulation of proinsulin inside the lumen of the ER. Intracellular disulfide bonds link together the N-terminal and C-terminal regions of proinsulin. Proinsulin is then transported to the Golgi system and to the secretory vesicles. Inside the secretory vesicles, insulin is subjected to proteolytic cleavage. The result is the production of active insulin formed by the N and C terminal regions that are connected by disulfide bonds. Therefore, insulin is formed by two chains called A and B. The region connecting the N and C terminal fragments and produced by proteolysis is a single polypeptide chain called a C-peptide. Both mature insulin and the C-peptide are then secreted by pancreatic cells. For the longest time, the C-peptide was considered necessary for the proper folding of proinsulin and insulin and was considered as an inactive byproduct of insulin processing once it was released. Recent studies have shown that the C-peptide has in fact many important physiological roles. For example, the C-peptide and mature insulin have opposing action on red blood cells. Red blood cells have an enzyme called PDE3 that converts ATP into AMP. When PDE3 is inactivated, excess ATP is released by red blood cells and triggers in endothelial cells the activation of eNOS, an enzyme responsible for the production of nitric oxide. The release of nitric oxide results in vasodilation. The binding of insulin to its receptor on red blood cells activates PDE3, while the binding of the C-peptide to its red blood cells receptor inhibits PDE3. Therefore, together, insulin and the C-peptide adjust the amount of ATP released by red blood cell and control the release of nitric oxide and vasodilation. In other cases, both insulin and the C-peptide have inhibitory effects. When white blood cells are activated, they release cytokines that trigger the upregulation of adhesion molecules at the surface of endothelial cells. The attachment of white blood cells to these adhesion molecules is one of the first steps of extravasation, the infiltration of inflamed tissues by white blood cells. When both insulin and C-peptides bind to their respective receptors at the surface of white blood cells, a signaling cascade is activated and leads to a decrease in the release of cytokines, and thus prevents tissue infiltration by white blood cells. Therefore, the C-peptides has anti-inflammatory properties. A model proposed that the C-peptide mediates the tuning of insulin signaling. Let's first see what happens when insulin level is low. The binding of the C-peptide to its membrane receptor leads to the activation of one type of G protein and the stimulation of an intracellular signaling pathway that mimics insulin signaling. Therefore, insulin signaling is enhanced by the C-peptide when insulin level is low. Now, what happens when the level of insulin is high? When the C-peptide binds to its receptor, a different G protein is activated. This switch of G protein leads to the activation of PKC, which in turn represses the insulin signaling pathway. Therefore, at high concentration of insulin, the C-peptide dampens the insulin signaling pathway. So in conclusion, protein processing is an important way to regulate protein activity. Some proteins are stored in a precursor and inactive form, and then when it is necessary, specific proteases cleave and activate the precursor. It allows cells to accumulate high concentration of inactive proteins and thus reduce their potential cell toxicity, and to quickly process and release large amount of the active form of the protein when needed.